All right, folks, it's 10 a.m., so we'll get going here. Good morning. My name is Meg Detloff. I work for PNAMP and StreamNet projects underneath Pacific States Marine Fisheries Commission. Thanks to everybody for being here today for our spring meeting of the PNAMP Fish Monitoring Work Group. Before we get going, I'd like to go over some housekeeping tips and tricks. If you guys haven't been to these meetings before or you are not familiar with Teams, this is kind of what the bar looks like. This is the chat icon. This is to raise your hand. And then these are to turn your mic and camera on and off. If you guys want to make sure you're muted and your cameras are off, it helps keep everybody focused during the meeting.
Do you want to go back to sharing, Meg?
Oh, shoot. Thanks, Mari.
You're doing great.
Thank you. No, I appreciate that. Thank you. All right. So now that you guys can actually see my screen, just so you know, we can mute you guys, but we can't unmute you. So if you do want to contribute to the conversation, you're going to have to unmute yourself. We're recording this meeting, and it'll be available on our YouTube channel next week. A couple of troubleshooting tips. If your Teams is being finicky, feel free to leave the meeting and then join. Sometimes that kind of helps wake Teams up. Also, try using a different browser. I mean, obviously, Microsoft loves Edge, so that usually helps your Teams instead of using Chrome if you're struggling there. And you can also try using your desktop instead of the browser version. Sometimes, or vice versa. Just kind of sometimes that helps if you're struggling. And sometimes too, some agencies don't allow the chat function. So if you guys don't have that option, obviously feel free to unmute yourself and ask questions. And with that, I would like to acknowledge the Fish Monitoring Work Group team. These folks have been part of our leadership on either one or more of these tasks on the right. We're grateful for their dedication to our collaborative mission. And also, if you can see the folks with the asterisks, that's our core team. And that consists of Casey Bleesner of ODF&W, Marika Dobos of IDF&G, Laura Erickson, Nancy Leonard, Mario Williams, and myself, all PSMFC, Russell Scranton with BPA, and Jen Bayer, who was previously the PNAMP coordinator but is now a contractor. So congrats to her on her retirement. And a little reminder that if you're interested in learning more about the Fish Monitoring Work Group or a particular task, these are obviously screenshots, so it's not super easy to read, but one of my wonderful core team will drop a link in the chat so you guys can access the project page, and that'll help you see basically some of the task pages, some of the related documents we have, and also links to previous meetings, which inside those meetings is usually a link to a YouTube video for the presentation if that was allowed by the agency or the person presenting. And with that, if you guys will open up the chat and type your name and organization, this helps us keep track of participation, but also it helps you acquaint yourself with the chat function. And then while you guys are doing that, I'll talk about the agenda. So basically, we kick off the meeting with task updates, and then today we'll have Zach and Mary present on lamprey that have been artificially propagated into the Tucannon River. So super cool. I saw them at the Pacific Lamprey Conservation Initiative meeting in December, and they were just really, really, really cool presentations. So I'm excited that we have them today. And then we're going to try a new open forum part after Mary and Zach's presentations. And if one of my team could mute whoever that is, that'd be super helpful. And with that, we'll get this going. So this section is tasking updates. We currently have 7 active tasks with 1 coming on board shortly. And I'll go through each of these, and some of my task leads will cover some of the tasks. And we'll just kind of give you an overview of what's happened since January. And then we'll talk about the open forum. And we're going to add some additional Q&A time for task updates, but we'll just kind of see how that goes and how chatty you guys are today. All right, and with that, in January, if you were here, I announced that we are nearing completion of the manuscript. Those following us know this has been thoughtfully developed. We've been thoughtfully developing this paper for several years. I'm so proud to announce that we will begin journal submission in the next week or two. We've completed internal review with NOAA, IDF&G, USGS, WDFW, US Forest, and DFO. If you're not familiar with DFO, that's Fisheries and Oceans Canada. Our first preference for journal submission is reviews in fish biology and fisheries, where we'll utilize open access to make the paper as broadly available as possible. And we are also planning on producing a best practices white paper as an additional resource. And I'll pause really, really briefly after each task. So if you guys have any burning questions right now, we can answer them, or feel free to type them in the chat and somebody will let me know that there's questions. Otherwise, we'll pause at the end after all the updates in case you guys need a little bit more time to marinate on a question. Any question? Right. And the next one, I'm going to hand this over to Sam Semino. He's with PSMFC. He's the lead on this task, which is actually an evolution of our rotary screw trap data collection task. And so,
yeah, thanks, Meg. So some of you are familiar with the rotary screw trap dashboard that started a few years ago, and it has evolved into something a little bigger and more user-friendly. We have expanded beyond just the Columbia River Basin. We have interest in Puget Sound and along the Oregon coast, and we're trying to get the Klamath Basin on board as well. But this is a data discovery tool similar to the salmon data discovery tool, if you're familiar with that, where the users can go into this tool and document where their juvenile fish traps are and some metadata associated with that. So we've been working with QW, our developers, on this, and we're hoping to have a product sometime in mid-summer to late summer. We are starting development, I believe, on May 4th. So it's coming to a kind of a conclusion for now, but yeah, we're excited about this, and it's going to be a useful tool for the public to see, or for the outside world to see where these traps might be located, and then it's going to be really useful for those who are operating these traps to document the metadata associated with it.
Excellent. Thanks, Sam. Any questions from anybody out there? Obviously, you can unmute and ask, or raise your hand, or type in the chat. Alrighty, excellent. Thanks, Sam. All right, get my computer to click. This is the pit tag data and analysis task. I'm currently gathering information to create a kind of one-stop shop for the pit tag, data and analysis resources such as Dart links or R codes for tools. I'm planning on reaching out to those on our distribution list for some suggestions. We've kind of built a little bit of a spreadsheet right now. We're also still determining, the follow-up from the webinar series last year, which was really popular. We're still trying to kind of figure out whether or not we can host an in-person workshop, kind of more focused on the analysis portion, or if it's just easier to create another web-based series, since travel and everything is a little bit tighter these days for folks. And so this is kind of a mock-up of the resource page will look like. I'm a biologist, not a programmer, so bear with me here. Hopefully this will be basically a button that you'll push that'll take you to a page with these tools and kind of some short descriptions about the tools with various other attributes. And then hopefully, stuff will link, or it'll just create kind of an easier access for folks to find resources, because I know there's a lot of stuff out there. And it's a little bit based off of the StreamNet resources page. So if you guys have been there, this is a resource page. And then when you click the button, basically, so I click the Northwest Fishes one, and it pops up into its own individual page with links and more information. So hopefully the one-stop shop will be something similar. We'll see how good I do at that. All right, any questions on that? Excellent. And
Hey, Meg, I was
yeah, go
trying to
ahead.
unmute.
Yeah, go ahead,
just,
Sam. Or, sorry.
No, it's Russ.
Russ.
Just for everybody out there, examples are things that you saw. We do have a list from all Dart tools. We have lists from tools like that have been presented in the past, like Pithy. We're trying to make sure that the updated tools for TBOM or other R-codes tools are identified. But if you have any other tools that you have published in R or available on GitHub that you think have some regional application, please reach out to Meg and me, and we'll make sure that they can get posted. The goal is to make sure that these hit tag analysis tools or tools that help people use hit tags and arrays are available. Some examples, or one other example, is that there are some codes that we have that document every single hit array order. So if you're trying to do an analysis of fish movement up and down streams, there's a file that gives you hit array order to and is being managed collectively rather than individually. So those are some examples of things that we're trying to get.
Awesome. Thanks, Russ. Yeah, that was great. Additional details on that one. All right, the focal species probable native range maps. The next task, basically Huck 12 maps for use by BPA CB Fish and Monterey Resources and other regional partners. The next slide, these are kind of draft examples, and we'll give you a flavor of the map functionality. So our first group of focal species is listed here on the left. We started with a sockeye pilot to kind of dial in the display format. And as you can see, these are broken into individual Huck 12 units. So the next slide kind of shows that when you hover over or click on one of these units, it gives you different attributes. And so hopefully, these attributes will help identify, displaying what species and how they've historically used these areas to help guide management decisions. And this is still just pretty brand new. So we're still working with our development team QW on it, but we'll be reaching out to some of you folks that are involved in some of these specific species to make sure that the maps are correct, that the information is correct. And so kind of expect that coming down the pipeline. And also, if you have any questions, feel free to reach out to me and I can direct you to resources or try to help give you more information. Any questions on that? Anything you want to, Russ? Do you want to add, Russ? Okay. I don't see you coming off of mute. All right, and the next one, I'll turn this over to Mari Williams of PSMFC, who will present the SAR-SAS task, and also the next one coming up too. Go ahead, Mari.
Thanks, Meg. So we are launching the evaluation group to review what the ISAB report contained with information for SAR versus SAS in coordinated assessments data. We will be starting that at the end of this month. And if you would like to join us or think someone else should join us, click on or use the QR code to go take the survey and let us know when you're available and how you would like to participate.
Awesome.
All right.
We'll make sure to add these links and stuff to the follow-up email so you folks don't have to scramble to grab your cell phones. Thanks, Mari. And Mari also gets the fun digital field guide too.
Yeah, so this is looking to create a digital repository field guide that can be used in chapters or chunks as necessary to update some of these older resources that are still being used today, but we have a lot of technology that has improved. And so our images and the way that we can share them is a lot easier now. So if you would like to be involved with this task, you can email myself or Sam Semino and Russ. Russ is really the the dreamer that has seen this towards actually happening. Anything to add?
Any questions? Go ahead, Russ.
I think, yeah, the main thing is we've had just some initial meetings on digital image documentation. While these field guides, the historic ones, are good, we met with Pitagis, and we're trying to link this to the Pitagis MR or run rear type codes methodology, so that people can have images. This field guide that you have as an example image has a image of a bull trout that's really shiny and poor lighting. We've talked with Stan Gregory and other people, and got other resources from online that have other images that may be better quality, but we're still looking for potential ways to document various fish species, whether it's juvenile bull trout, adult bull trout, mixed species, invasive species. We're just trying to improve those resources for those images and give credit to the people who have those photos. And so if you have images and you'd like to share them, we'll be giving some more updates in the future on that.
Yeah, and we are looking to crosswalk with existing programs. There is no interest or drive to create a whole unique set of ID codes, etc. for this project. So alignment with Pitagis codes for species run rear.
Thanks, great info, guys. Any questions? And sorry, I skipped over questions for the SAR/SAS task. So
that's okay. We've got space at the end too. So
yeah, we do, and we're nearing the end. Laura, I think you're probably out there. So Laura Erickson of PSMFC is gonna talk about this next task that we just initiated.
Yeah, so this one is we're identifying some of the genetic fields that potentially could be added to the ARMIS data system. And this will just enhance access to genetic data among our member states. This is one of the resolutions that Pacific States has from our annual meeting with our member states. IDF&G is certainly further along with PBT and work, and so we're collaborating with them and others. California is bringing a lot of their work online as well. So we're just looking at a way to leverage the existing coded wire tag system and potentially look at a way to have a shared database among our member states and across agencies. And then we do have a link at the bottom. I've also shared it in the chat. If you're interested in being part of it, we're looking to hold a couple of workshops in the next couple of months. And so if you'd like to participate, please fill out the form that's both in the link on the slide deck as well as in the chat. And Mario or myself will reach out to you and make sure that we include you in the discussions. Yeah.
Excellent.
Thanks, Laura.
Thanks, Laura. Any questions from anybody? And if not, we're about to go to the. Do you have any questions on any of the tasks? So, all right. Thanks for dropping those links. Okay, so this is where we pause in case you guys have been marinating on something and want to ask any questions or get some clarification. I'll just kind of pause here for a minute. You guys are shy today. All right, going once.
We're all just excited for the Lamprey talks.
I know, me too. All right, so let's get into this. Like I mentioned earlier, I actually saw these talks at the Lamprey Information Conference in December, or the Information Exchange. And Zach and Mary were absolutely brilliant. So I'm super excited we have them here today. Zach's going to present first. He's a lab manager and biologist of the Pacific Lamprey Research and Restoration Project with the Confederated Tribes of Umatilla Indian Reservation. And then after Zach's presentation on the Tucannon Lamprey restoration, Mary's going to talk about how they basically used the artificially propagated larval lamprey in the Tucannon and kind of all of the ups and downs of that. And Mary is a PhD fisheries biologist with Moserworks LLC now, and she was previously with the National Marine Fisheries Service. So with that, I will hand it over to Zach. I will stop sharing my screen. All right, perfect. Looking good, Zach.
You all hear me?
Yep, sounds great.
All right. Thanks, Meg, and thanks everyone for having us here today. Let's see. Today I'm going to talk or present some preliminary results from a pilot study that's been carried out by the Confederated Tribes of the Umatilla Indian Reservation with assistance or really leadership from Mary Moser of Moserworks LLC. And the genetic components of what I'm going to present have all been, the heavy lifting on the genetic stuff has all been done by John Hess and Greg Silver and others at CritFic. And I want to emphasize that I'm running this presentation. I'm a Johnny come lately to this whole thing. Really, the masterminds behind this project were Aaron Jackson, Mary Moser, Alexa Main, and John Hess, I think. So let's get into it. Since I think 2021, we have propagated and released approximately 4.3 million larval lamprey in the Tucannon River, and we've been using a technique that I think this audience is hopefully pretty familiar with, called parentage-based tagging, to monitor them in the wild. If you're not familiar, parentage-based tagging is a technique that allows us to use genetics to conduct mark recapture surveys of our animals. And before I move on, I want to add one important point of emphasis, which is that here, artificial propagation of lamprey is not being proposed as a solution. We're just highlighting a unique system in the Tucannon River that was identified as a good place for testing these methods and application. And I accidentally hit my keyboard or something, but that arrow is just showing one of our release sites on the Tucannon River there. Don't really need to remember this, but I thought it was kind of cool. That is, you'll see later on in the results, that's an area where I think it was identified as Index 12. Quick note on some abbreviations. I may default to referring to Confederated Tribes of the Umatilla Indian Reservation as CTUIR, Columbia Intertribal Fish Commission as CritFic, and Artificial Propagation as AP. So I always like to start by emphasizing that Pacific lamprey, also known as Casuyas in the Umatilla Sahaptin dialect, are a first food for CTUIR as well as the other Columbia Basin tribes. That's really the whole driving force behind all of this work. Here you can see some photos, historic photos of people catching and consuming lamprey in the Columbia Basin. And within the CTUIR, Department of Natural Resources sort of organizational structure, lamprey are managed by a fisheries program, which makes a lot of sense. But I highlight that because the mission of the CTUIR fisheries program is to establish and maintain sustainable harvest opportunities of all of their traditional subsistence foods or first foods, aquatic first foods in particular for the fisheries program, and that includes lamprey. This is a little bit of background on the life cycle of lamprey. They're similar to salmon, but not exactly the same. It shows kind of some of the time periods. It's a lot more variable for lamprey than it is for salmon. You see larvae transform into juveniles anywhere in the first 13 years. Right there. And the other thing I wanted to emphasize is that one of the key differences between lamprey and salmon is that whereas salmon typically return to the same spot in the river where they were born, lamprey don't necessarily do that. The adults are queuing in to pheromones being released by the larvae in the river. So they theoretically could return to any river where they're detecting pheromones being released by the larvae. And I'll refer back to that later on in this presentation. This is a map of the Columbia and Snake rivers, and it shows where the Tucannon River flows into the Snake River in between Lower Monumental and Little Goose dams. It's the most downstream tributary to the Snake River that sustains populations of anadromous fish, including lamprey in the lower basin, and now more recently in the upper basin in Tucannon. And I've highlighted Ice Harbor Dam in this map because it refers to the next slide, which shows kind of history of dam passage for Pacific lamprey at Bonneville Dam in the orange, and Ice Harbor Dam in the blue. Ice Harbor Dam construction was completed in 1962, and you can see that first year they had over 36,000 fish pass Ice Harbor, and then after that it plummeted and it really hasn't recovered. And for some reason they stopped monitoring right around 1968, 1969. Didn't start monitoring again until 1999. There's something else I was going to say, but I don't remember. Oh, this was the first major impact to Pacific lamprey on the Tucannon. The next major impact was shortly after the construction of Ice Harbor Dam. The largest flood of record on the Tucannon River happened kind of over New Year's in December 1964 and January 1965. And response to that from local landowners and the U.S. Army Corps was they were tired of the river trashing their production ground. Tucannon is largely an agricultural river, a lot of farming and ranching, and so you can see here landowners going in and they honed out the channel. They just reamed it and straightened it, which significantly reduced available habitat and impaired the river's ability to establish new habitat, because it basically just turned into a water conveyance ditch that wasn't able to accumulate the fine sediments that larval lamprey are dependent on for habitat. And you can imagine that any benthic organism is not going to do well when there are bulldozers scraping up the stream bed. So the general consensus, I used to work on river restoration projects on the Tucannon for the tribe. My conversations with local landowners and fish biologists, people who had lived on the river for over 70 years, the general consensus is that lamprey have largely been eliminated from the upper river. And Mary Moser, along with David Close from the Umatilla tribes, published a study back in 2003 where they surveyed for larval lamprey in all 5 of the major watersheds in CTOIR's historic territory. And here in this map, you can see the black dots are showing places in each river where they detected larval lamprey, and the white dots are showing places where they did not detect larval lamprey using electrofishing methods. You can see even in 2003, they did not detect larval lamprey in the upper Tucannon. So in response to all those white dots on the previous map, CTOIR got together with Yakima Nation, Fitfic, and the Nez Perce tribe, and they developed this master plan for artificial propagation, translocation, to return lamprey to their historic territory. And then as a part of that master plan, it also included setting up research to test both of these restoration methods and compare and contrast and see how each one works for reestablishing larval lamprey in their historic territory. In the master plan, the Tucannon River was specifically identified for the release of 1 million larval lamprey annually. That's the goal every year. We just started spawning in the lab right now. We're probably going to spawn today. So the research objectives for artificial propagation and release in the Tucannon are as follows. First was to confirm whether or not artificially propagated larval lamprey can actually survive in the wild, and for how long. If they do survive, can we assess growth and downstream distribution? And then to see basically if we can actually use parentage-based tagging to trap larvae and familial groups in the wild to match, and then, as Mary's going to talk about later, match to some of the experimental treatments that were tested. This is a quick overview of the spawning process. The adult broodstock are collected at the 3 lower Columbia River dams listed here. We usually get a subsample of those, about 50 adult broodstock or less, back at the lab every year. They overwinter in the lab, and in the spring, right about exactly now, we have a bunch of females in the broodstock tank that look like the picture you see there, a gravid female. They reach sexual maturity, and then we strip the eggs. We keep this is an important point of emphasis, we keep the eggs of each female separate. That allows us to establish matrilineal family groups and then track each family group through the female genetics. And then we combine milk from all the males and then divvy out equal portions of the combined milk to each female that we were able to get eggs from in a given spawning attempt. That kind of helps us maximize some genetic diversity in our methods. These are some of the stages of development after the eggs have been fertilized. Important point of emphasis here is we try to target them for release right around the time that you can see that yellow strip in the abdomen of that larval lamprey. That's a yolk sac that's slowly turning into their digestive tract, and as the digestive tract connects with their anus, that means they're ready to start feeding through their mouth in the wild, and that's what we target for release into the Tucannon. This is, I'm going to cover this very high level. This is basically Mary's presentation. Tested 2 different release methods. The first was putting individual family groups into these plastic containers you can see in the picture, and then release them at staggered intervals of 6, 24 hours, 3 days, 5 days. Boom. The other release method was we put individual family groups into buckets and release them either into slow water or fast water directly into the river. So now for the results. Starting in 2020, our lamprey crew went out and conducted electrofishing surveys at each of the index sites that you see here. And consistent with the results from Moser and Close 2003, that map I showed earlier, they didn't find any lamprey in the upper Tucannon. They only found lamprey in the lower Tucannon, and this is all prior to any releases that were by our group. So they were only detecting wild fish, and they were down in the lower river. Now, we think one of the limitations, potential limiting factors in the Tucannon, is right around that red dashed line. There's a geologic feature called Tucannon Falls. It's basically just a bedrock cascade, rapid, and for whatever reason, there aren't a lot of adults that go past that. So these are the results from 2020. No assignment in gray in these next several pie charts means we detected larval lamprey, but we were not able to assign any known genotype or genetics to the larvae that were collected. And in this particular case, in the lower basin, that's because those are wild fish, and we don't have the genetic profile for those fish. So in 2021, we released about 470,000 larvae at the sites in the index sites 10 and 11 highlighted here. And when our crew went out and conducted electrofishing surveys, they didn't detect any larval lamprey in the upper Tucannon, but they continued to detect wild fish in the lower Tucannon. 2022, we released about 380,000 larval lamprey, again at index 10 and 11. And our team started detecting fish, but we don't currently have the genetic results back for that, but we're pretty confident that those are our artificially propagated fish, because we started genotyping them in later years, and it's almost exclusively artificially propagated fish. In 2023, we added 2 new direct release sites at a place called Blue Lake and a new index site called Index 12. In addition to Index 10 and 11, and we released 950,000 larval lamprey. Don't have the genetic results back for that yet, but continued to detect larval lamprey at all of the release sites. 2024 added 1 more direct release site at Index 6, down lower in the watershed, and continued all the other release sites, and we released 1.4 million larval lamprey. We have the genetic results back for the surveys, the post-release surveys that were conducted in that year. And we started identifying some of the artificially propagated larvae from previous release years. You can see here we have fish from brood year 21 and 22. And some unassigned fish still down at index 11 and 12. And this is the most exciting slide. We're starting to see fish from 2023. And then you can see there's a new slice of pie down there in index 1 through 5. And those fish were not actually electrofished. That's representative of about 1,500 larval lamprey that were collected at that black star in the upper left at a screw trap operated by WDFW. And that slice of pie represents about 2 or 3 dozen of our brood year 21 fish that were collected at the screw trap. I believe that's the first year that they've collected and identified fish from our artificial propagation. This is really high level, just basic representation of the size classes of the different samples. You can see that the screw trap samples are all, the fish are much larger than the fish from the same age class, brood year 2021, that were sampled up at the upper index sites. But at this point, it's unclear whether that's a real signal or it's just that the screw traps are unable to capture those smaller fish. We also, so lamprey go through a metamorphosis from these eyeless filter feeding larvae into a miniature version of the adults with eyes and a sucker mouth. And they did capture at least 1 transformed juvenile at the screw trap that was from our brood year 2021. So the main takeaway points here is just the means in the middle of each box and whisker, and the fact that there's no real overlap between the index sites and the screw trap samples in those little whiskers, which suggests, at least looked at the data in this way, they are significantly different in size. Back in January, I had a chance to go out with the survey crew, and this is one of our larval lamprey collected way up at index 10. Judging from the size, it's maybe brood year 21 or 22. So getting back to those research objectives, we have detected Pacific lamprey, artificially propagated Pacific lamprey that have survived up to 4 years from all of our age classes. John Hess always wants me to emphasize that this is some of the most precise age data that we've collected in field samples of lamprey. We do know the age down to the hour of fertilization, which should hopefully, as this project continues to grow and we collect more data, allow us to really gain some really fine level information about life history of lamprey. The AP larvae that were collected at the upper index sites grew at an average of about 33 or 30 millimeters a year. Then the fish that were collected at the screw trap grew at about 40 millimeters a year, just taking average size and dividing it by the number of years they've been in the river. AP larvae have been detected up to 50 kilometers downstream of their release site. Here's based on the screw trap samples that these age 4 larvae are now leaving the watershed. And we detected that one juvenile. But wait, there's more. We had a bonus objective, which was not in our plans. There's some very anecdotal evidence that perhaps these larval lamprey might be attracting adult lamprey into the upper Tucannon for the first time in 70 years. If you recall, I mentioned that the adults hone into where the larvae are releasing pheromones as a signal of appropriate places to spawn. And if there aren't any larvae, then there's not going to be that signal for the adults. This is an email that I received from a U.S. Forest Service fish biologist back in 2023, 2 years after the release of the first artificially propagated lamprey on the Tucannon. And he says he saw them way upstream, even upstream of our release sites. And this is a career fish biologist for the Forest Service saying he's never seen adults that high in the system before. So that's Panjab Creek. That's the creek that Bill's referring to in his email. That's where Bill saw his adult lamprey observation. And in the next slide, I'm going to show a video that he took, trying to capture this, evidence of this adult lamprey way upstream. I don't know if the audio is going to come through, but at some point he says, "Look, there's a lamprey." So here, there's that gray thing going up from top to bottom or bottom to top on the stream bottom. That's a pit tag array. That's just purely coincidental. These fish are not pit tagged. It just happened to attach to a pit tag array. And then in the middle of the screen, you can see kind of hanging downstream from middle to the left. If I play the video, you might be able to see a lamprey attached to that pit tag array, kind of swinging around in the current. So I would like to make the argument or posit that our larval lamprey are attracting adult lamprey into the upper Tucannon for the first time in 70 years. Now on to the next.
Awesome. So cool, Zach. We can pause just for a second as Mary's kind of transitioning. Did anybody have any hot questions they want to ask? Otherwise, we can pause that.
Evan has a question in the chat.
Yeah, go ahead, Evan.
Howdy, folks. Hey, I was just wondering, were they, or can WDFW pit tag the presumed migrants moving past the screw trap?
I don't know what WDFW's capacity is, but you're not the first person to ask that question. It would probably be a question of funding and coordination between Aaron Jackson, the Lamprey project lead, and whoever's running the screw trap.
Go ahead, Michael.
Yeah, so actually, that's out of my office in Dayton. When we were first contacted about taking genetic samples, we asked about pit tagging, but I think a lot of the limitations were due to the fact that the fish we're capturing at the smoke trap don't meet the size requirements for pit tagging. And so that we haven't pit tagged them at this time.
Thank you.
Excellent. Good information, Michael. All right. And we'll have another moment to pause after Mary's presentation for more questions. So go ahead, Mary, take it away.
Okay. Can you see my presentation okay?
Yep, looks great.
All right, good. Well, yeah, thanks for having me. And thank you, Zach, for such a great lead into my talk. As he mentioned before, I'm just going to be talking about more of the specifics of what we're testing with best methods for releasing artificially propagated lamprey. And my thing won't advance. Let's see. That is, oh, there we go. Okay. So we aren't the first ones to ever release artificially propagated lamprey into natural reservoir systems. They've done it in Finland and in Japan. And the typical method is kind of what Zach mentioned before, this direct release method, or basically dumping from a bucket. And so we wanted to test and see whether, number one, whether fish were surviving that experience. And obviously, we know now that they are because we have this genetic work. But back in 2021, we wanted to make sure that the fish weren't just dying as soon as we put them out. So that kind of led to this 5-year study of survival after release into these artificial red chambers. And so Zach kind of pointed these out. These are 10-liter polycarbonate pans that we seal the larvae into. And then those little circles on the top are fine mesh screen that prevent the larvae from getting out of the chambers and allow us to look at survival at 6 hours, 24 hours, 3 days, and 5 days after the release. So we had several hypotheses that we wanted to test. One was that these volitional releases would do better than direct releases just because the lamprey could go out on their own time. What we do is rip off the top screen on these chambers, and that allows them to go out through a larger screen but still avoid predation. So that was our first hypothesis. Our second hypothesis was that pre-acclimating them to Tucannon river water would help survival. And we were worried about this because early on, we found that these first feeding larvae are really sensitive to water quality changes. And we were worried that if we reared them and incubated them in well water and then just dumped them out into the Tucannon, that that water quality change would be too much for them. So we wanted to test that idea. And the way we did it was we actually had 2 identical recirculating rack systems that we incubated the separate family groups. Zach mentioned that we keep all of our treatments separate by the female genetics. So we are able to trace back to the exact treatment that they came from. And then our third hypothesis was that older larvae would be able to survive better than younger larvae. And the window we're looking at here is pretty small. So we're releasing them before they're completely onto exogenous feeding. So these guys are 23 to 40 days after fertilization. And the one on the left there is a 23-day-old fish. You can see, as Zach mentioned, the yolk sac is really well developed. It's still there. It's feeding on endogenous stores still. And then the one on the right is a 40-day-old larvae. And you can see the yolk sac is almost all gone, and it's much more well developed. The eye is really starting to be noticeable. You can see the much more developed branchial basket, and just it looks like it's much more together than that other fish. So kind of just some numbers. Zach alluded to sort of the development of our program in 2021. The first year, we didn't have very many brood stock, and we were able to release releases for about 470,000 fish. Of those, about a quarter went into those artificial red boxes. And then we had kind of similar results in 2022. We realized that we needed to have more brood stock. So Aaron Jackson, who is the mastermind of this whole program, arranged for there to be more brood stock for us to use. And so then we were able to reach close to the 1 million mark in 2023. And so as you can see, the percentage of larvae that were went into the direct release versus the artificial red boxes changes over time. But we had kind of similar numbers in those artificial reds over time to look at our hypotheses. It's not it's stuck again for some reason. It's so weird. Let me see. I have a feeling it's just my bandwidth is the problem here. So I guess patience is a virtue. Huh.
It's all right. We can all take a moment to breathe.
Take
Mary.
a moment. That's right.
You're doing great.
I keep pushing the button like, come on, come on. All right. Well, I'm not sure what to do. But anyway, I can move forward with the results.
Do you want me to put yours up, Mary, and run it?
Sure, yeah, if you can do that in the background while I'm waiting for this to come on, and if it doesn't, then if you can get to this slide on the PDF, that'd be awesome. Sorry about this.
No, you're totally fine.
This gets to me all the time. Here I am in Seattle. Like, I live in Seattle, and I have the worst bandwidth anywhere. I can be in the boonies of Eastern Washington, and I have better service than I do here. It's so pathetic, but yeah, it's not. I don't think it's gonna go. So I'm gonna stop sharing, okay?
Yeah, that's okay. Here, I will share my screen.
See, it's good that we were thinking so far ahead here that we were able to pivot. So could you go to the next slide, please? Thank you.
Yep, totally.
Sorry about this. So okay, so that's great. So this is Alexa, Maine. This is on our big first day when we're releasing fish. These were actually photos from last year, and we're putting the artificial reds out. And the way that we assess the survival is that at those 4 time periods, we take a subsample of larvae in a beaker and we take a video of that. And then we use a still shot to actually get a count of the number of larvae. And then we use the video to assess how many of them are alive. So that is the method that we use for coming up with these survival estimates. Okay, next slide, please. All right, and keep going. There's the results. Okay, so the first hypothesis that we were assessing, well, I guess I didn't mention this one, but we were curious about whether survival in those red boxes would improve with time. And so these are the results from the 5 years of study combined. And you can see that we had like 97% survival after 6 hours. But that drops down to about 80% survival at the 5-day period, which is when we sort of empty all the chambers and our experiment. So you can see that the hypothesis was not upheld. In fact, their survival decreases with time. And can you go to the next slide, please? And we have now some knowledge about why that was going on. These are results from last year, and the dark dots show the dissolved oxygen. Starting in 2023, we started measuring dissolved oxygen in the chambers over time. And what we found was that because of siltation, those screens get blocked, and then there's not as much water exchange in the chambers. And we've got about 10,000 larvae per chamber in there. So after 5 days, the dissolved oxygen was getting down to hypoxic levels. It's in this case, it was around 22% at the 5-day period, and we start seeing increased mortality. So a couple things that came from this. We decided that we're no longer going to be running these artificial red chambers for longer than 24 hours just to obviate problems with hypoxia in the chamber. And also, this almost certainly is the reason that we had that reduced mortality over time. Okay, next slide, please. All right, the next thing we wanted to test is whether the pre-acclimation to Tucannon water resulted in higher survival. And again, this is the combined results over all of the years. And you can see that there was no obvious benefit that was happening from that, which is really great for us because bringing Tucannon water into the lab is just a biosecurity nightmare for us. And so it's good. It's good that the larvae are tough enough to be able to just be transferred directly from well water incubation to Tucannon water. One thing is that exposure to that Tucannon water microbiome early in their life may convey some later advantage, but I'll speak to that in a minute with the genetic results. Okay, next slide, please. Okay, and then there was this one about whether the younger larvae were surviving better than, or older larvae were surviving better than younger. So we just split. We had a actually pretty even split in treatments between those that were 30 days and younger and those that are 30 days and older. So again, these are small windows. So 23 to 30 days, 30 to 40 days are the two groups. And you can see that the older larvae did survive slightly better than the younger larvae. But again, my feeling is that this was a consequence of the hypoxia problem because these little guys, when they're less than 30 days, they're not very mobile. They're not very adept. I mean, they will eventually get out of the chambers, but these older guys, as soon as we take off that small screen, they're out of there. They can quickly exit the chambers. And so I think part of the reason we're seeing this lower survival of the younger larvae is because they were exposed to hypoxic conditions in the chamber for longer. One other thing is that when we release these, when we do the direct releases of these really young larvae, they look like snow in the water. I mean, they're pretty visible because of the very large yolk sac. And I think it probably makes them more of a target for predation. So that would be another reason to go with older larvae. Okay, next slide, please. And then, as Zach mentioned, we've just had a few larval collections that we have genetic results from. John Hess has been great at providing us with information from these, and it's just going to get better and better as time goes on. Right now, we have relatively low sample sizes, but it's encouraging because they all sort of back up what we've seen in our experimentation. And one thing is that we've seen larvae, electrofish larvae that we can trace back to both volitional and direct release methods. So the fish that are just dumped into the river are surviving. Eventually we'll be able to compare the relative percentages of the direct release fish to the percentage that were released, but we see samples of fish from both of those methods in the genetic results from fish that were collected in 2023 and 2025. We also see fish from both the pre-acclimation Tucannon water incubation and the well water incubation, and also examples of fish from larvae released at ages that were less than 30 days and over 30 days. So this sort of mirrors what we've seen in the experimental results, and that was encouraging. And we just can't even really do statistics on this yet, but soon we'll have enough genetic results to be able to do that. Okay, next slide, please. I think this is my close to the last one. Just to reiterate, the genetic evidence indicates that the direct and the volitional methods work. The direct method is way easier, and it's a pain in the neck to load all those chambers and then make sure that the larvae stay alive. We actually camp out there and guard them when this is going on. So I think our results are indicating that this direct method would be fine, and you can dump them and they'll do just as well. But it's been really nice having the volitional release work to use. And one thing we've talked about we may do this year is do the direct release at night so that the larvae have a little better chance to escape predation. Again, the larval mortality increased with time, probably due to that hypoxia problem. So we're not doing that anymore. And the pre-acclimation doesn't seem to convey any advantage, and we're not doing that anymore to avoid the biosecurity problems. And finally, the 30 to 40-day-old larvae were able to escape the reds better. They burrow, they're just more with it altogether. So we're also really focusing on doing first feeding larvae that are close to the end of their endogenous stores. And next slide, I just want to acknowledge the huge number of people that have helped out on this project. The funding came from BPA. Thank you, BPA. The staff of the Water Environmental Center at Walla Walla Community College, at the Imtwaha Hatchery, at Oregon Zoo, and High Desert Museum all helped us with broodstock maintenance. We don't put all of our fish eggs in one basket, and so that has been a huge benefit. Julie Burke from the Umatilla tribe is great administrative support. And we have a huge number of Whitman College student interns that have helped us over the years to maintain the fish in the lab. And finally, Sally G from Oregon Department of Fish and Wildlife gives us health screening results so that we can screen our larvae before they go out. We also wanted to thank Hagerman Laboratory, and particularly John Hess and Greg Silver, for all the amazing genetic results, and also the Yakima Nation. Ralph Lampman has provided us with reproductively ready adult lamprey when we really need them. So thank you, Ralph, for doing that, and I'll be happy to take any questions.
Excellent. Awesome. Stop sharing and turn my camera on. Don't turn your camera on, Mary. You don't have any Wi-Fi.
I know. Oh, it's so pathetic. Oh my gosh.
All right. Any questions?
At least my
anybody?
voice. At least some. Of course, I guess I could call you guys, and that would work too. But yeah.
No. Yeah, at least that's coming through. That's all that really matters. I'll get us started. I did have a question. So what do you think made that particular area of the Tucannon ideal for this experiment?
Well, there were several things. As Zach mentioned, lamprey had been extirpated up there for a very long time, and so it was easy to track, kind of the progress of these artificially propagated fish more easily. I failed to mention that Aaron Jackson also takes eDNA samples of the water before and after this program got started. And so those they have also kind of mirrored the progress that we've seen with fish moving downstream as the artificial propagation has gone on. So having that sort of empty lamprey situation, and really it's nice having the fact that there's some wild production in the lower part of the drainage, so we can actually see this proportion of the artificially propagation that's, contributing to the population. So that was kind of a little bonus aspect to it. And the thing that Zach mentioned with the adults being attracted back up in there, I mean, that would have been impossible to see if there had been production up there previously. But I think that's really good evidence that this is a, kind of an added bonus of the artificial propagation is having that pheromone cue to attract wild adults up there. So yeah.
Yeah, very cool. I agree. That's super cool. Has anybody else approached you about trying this in their systems, or?
Well, in the plan that Zach put up, the next system that is targeted is the Walla Walla, and in the master plan for supplementation, the idea in the Walla Walla was to try releasing fewer numbers of older larvae, that are not just at this first feeding stage, with the idea that they may have higher survival. And but we'll see how that. I mean, larval lamprey culture takes a huge amount of space, and being able, as you see here in the plan, thank you, Zach, that's awesome. You can see the Walla Walla number, the target there is 500,000. We need a much, much larger facility to be able to produce older larvae at those kind of numbers. So we're working on it. There is a plan to potentially have space at the Mtwaha hatchery to help accommodate this, rearing of larger lamprey. But our existing facilities just are, we're just barely able to accommodate the 1 million first feeding size ones. So yeah, it's weird to think about, but lamprey are very two-dimensional. They are in a plane. They're not taking advantage of all the water column, and so the aerial extent that you need is really large for lamprey culture. Yeah.
Excellent. Anybody else have questions? You can raise your
A
hand.
couple
You can type it in
chats
chat.
couple questions in the chat. Rich Townsend asks, was there any rehabilitation to the Tucannon River environment during this period as well?
I'll pass this one to Zach because he was actually involved in the restoration projects there.
Yeah, there's been some pretty intensive restoration going on for over 10 years now on the Tucannon River. From what I understand, the Tucannon River, because of its strategic location, hold on a second, that never happened. Solicitor calling. Holy moly, that was annoying. Okay. Yeah, BPA has targeted the Tucannon as one of its strategic watersheds for salmon restoration, kind of because of where it's geographically located in the Lower Snake Basin, and the fact that I don't know if I'm going to get this exactly right, but it's one of the only endemic wild species of Chinook or runs of Chinook left in the Columbia Basin, I think. So they have set up a programmatic pot of funding for restoration starting all the way back to 2010 or earlier. So they've been doing a lot of restoration, and the upper section is almost entirely on public ground owned by WDFW and the U.S. Forest Service. So there hasn't been that additional hoop to jump through of finding willing landowners. So yeah, they've done miles of restoration, and they're going to go back through and do more now because the Umatilla tribes and the Nez Perce tribe and WDFW are all collaborating on expanding restoration up there because there's a bunch of infrastructure that needs updates. And so they're going to use that opportunity to go in and improve conditions for fish habitat as well.
Thanks. Great question. And I see another question. How long do the lamprey release? How long do the larvae release the adult attracting pheromone after they're released?
Yeah, I can answer that one. So the pheromone is actually a bile acid that the larvae produce their entire larval existence. So from the time that they start feeding all the way until they metamorphose, they produce this bile acid, which is attractive to the adults. So yeah, I mean, so your signal is just getting, as you're accumulating more lamprey in the system, your signal is just getting, bigger and bigger over time.
Such a cool species. Anne says, great presentation. Thank you. Given the large footprint involved with the culturing lamprey, are there any plans to release translocated adults in the upper Tucannon to compare comparative survival of artificially propagated versus translocated progeny?
Yeah, I can address this too, because this was actually in the original, when the Umatilla tribe was first kind of making their strategy for how to assess artificial propagation relative to translocation. There was a conscious effort to keep the basin separate. So the John Day system is one that they decided to just leave. That's the control system that we would be comparing to. And then in the Umatilla and I think the Grand Ronde were the two systems that would have translocation. And then the Walla Walla and the Tucannon, also in the Umatilla area, is the two systems that were targeted for artificial propagation. So the strategy that the tribe has is for, oh, thanks, there it is again. This is, I had to get a copy of this slide, I guess, but yeah, so you can see here's the sort of, strategic plan is translocation in Upper Grand Ronde, Catherine Creek, Looking Glass, and then the John Day and the Imnaha are where the controls are. So in answer to your question, Anne, we are not doing translocation anywhere that we're doing artificial propagation to kind of keep those separate. Now, the Yakima Nation has a really different strategy, and I think it'll be really interesting to compare because they are doing both artificial propagation and translocation in the same areas, and then using the genetics to make those comparisons. So it's kind of just a whole different strategy. Yeah.
Excellent. Those are great questions, everyone. All right. Well, I think everybody's winding down. All right. Thanks, Zach and Mary. That was a fabulous tag team presentation about a really cool, kind of unique experiment that you guys are conducting, which I'm happy that it's being successful and that we're getting some really cool results. So that's very fun.
Yeah, thank you.
All right. And with that, I will take the screen over. And okay. All right. So this is what I mentioned earlier. We're introducing a new kind of open forum. So this is a time for you to share information with your colleagues about new publications, ask questions or request for information. We can try to hook you up with someone who can help you out if you have specific questions. You can recommend presentations for the group, or just a general update that you think would be of interest to this particular group. And just to kind of help kick this off so you guys aren't super intimidated by this new section. I this kind of over the past week has built and built. So apologize for it being a little overwhelming, but at least you get a flavor for kind of everything that you could possibly talk about. So as I mentioned earlier, the Pacific Lamprey Conservation Initiative, really great group of folks, and the Bonneville Power Administration. The FY27 request for proposals is out now, and I think maybe some of my core team can drop some links for you guys, and I'll provide these again in the follow-up email. So in case you have interest in any of this stuff, you can kind of have those links. Also, the PLCI has launched a new interactive story map titled Pacific Lamprey, Our Ancient Friend. I took a look at it. Very cool. Lots of history, lots of information, lots of cool pictures. Highly recommend you check it out. Also, the Pacific Salmon Commission released the technical report number 58, the specifications and definitions for the exchange of coded wire tag data for the North American Pacific coast. Mariel dropped that link too. So new tech report out for you folks to enjoy. Also, come work with us. PSMFC is seeking a database management specialist for Klamath Basin specific data architect and engineer out of Klamath Falls. We'll drop the link to that. Also, Bonneville Power Administration is seeking a Fish and Wildlife Administrator for the Portland office. And also, the Pacific Salmon Commission is seeking an assistant quantitative biologist for Vancouver, BC, in case you want to move to that beautiful area. So Mari will get those all posted. And then also, if you haven't heard of it, they have... What was that, Laura?
I was going to say we're seeking a quantitative scientist as well.
Okay, cool. So if you guys head to the PSMFC web site and the careers page, there's quite a bit of job openings. Nancy just wanted me to highlight that one, but like Laura said, they're hiring as well. We're hiring a bunch of seasonal positions kind of all over the partner states. So in case you guys want to pass those along, or if you have some interest in any of that, if you're new bios and kind of want to dip your toes into some different work. Also, so emerging technologies information sessions. We recently opened the abstract and poster submission, and so there'll be a link for that too. That'll kind of give you more information about those sessions. They're really neat. We've done webinar series. We've hosted previous workshops or conferences, apologies. And so that will be in Skamania this year in October. So we're kind of getting ready for that. That's really exciting. And then lastly, on my little information for you guys, the Klamath Basin Fisheries Collaborative Annual Meeting is next month on May 19th in Klamath Falls. The registration and agenda are coming soon, but they are accepting presentation or discussion topics. And if you're not familiar with the KBFC, they bring together those using pit tag technology for fisheries monitoring, research, and management in the Klamath Basin. So if you guys want to pass the word on that, if you have colleagues down there, that's usually a pretty good meeting, super informative, a lot of good folks in that group. And with that, I'll just kind of open it up. And Laura posted the PSMFC careers link. Thanks, Laura. And you guys can obviously post links in the chat if there's any publications you want to share. And I realize this is new, so you may have not come prepared, which is totally fine. So it's just kind of an opportunity for you guys to share or suggest anything to me I can track down. I'll just kind of give it a moment. I'll give you a moment of silence.
Thanks, Meg.
Yeah, sure. Do you have any updates, Russ, that you want to share?
No, you covered it. I think part of this is also that, just as you said, if you have new emerging technologies, new reports and publications, something that you want to share that could help others do their job, or just might be a question for future discussion, or might be a opportunity for a future presentation like we just had. Like people in the community to speak up or share that information with us.
Yep, absolutely. We're all about collaboration and communication here, so we're a resource for y'all.
Hey Meg, I can share something.
Yay, Casey, go for it.
I was just going to share that there's a special topic in Frontiers in Ecology and Evolution on intensively monitored watersheds. And a number of us in the fish monitoring work group have authored or co-authored papers in that. I'll drop that special issue in the chat. It's all open access. So if you're got any interest in intensively in IMW work, it's pretty great. There's, I think, 10 or more articles already published in that edition.
Awesome. That's a great plug, Casey. Also, I'm thinking too, if you guys aren't familiar, the Western Division AFS is actually in Portland this year. And there's going to be a lot of great sessions and all kinds of fun new presentations. And I think we're hosting 2 sessions.
Yeah, Mari and I have a data focus session that we got enough abstracts to have a full day. So if you're at AFS and want to come hear about data management, AI, data technology, come see us on Wednesday.
And it will be on top of the effectiveness monitoring session that a lot of the papers that Casey just pointed out will be reporting on.
I'll be torn. Oh no.
Yes, it will be hard to pick what to actually get to go to. Obviously, I'll be moderating.
Yeah, you're stuck in your session.
Yeah.
Nicole, I see you're off mute. Did you want to contribute anything?
Yeah, I just wanted to let everybody know that the pit tag steering committee just released an update to the pit tag marking procedures manual that has been published for decades now. And it's a general update of all the information, but in particular includes new information on marking smaller fish with 9mm tags and also marking lamprey with pit tags. So I can throw a link in the chat, but that was just recently published last month.
Awesome. That's a great yeah, I'll steal that too and put that in the PNAMP newsletter because that's really cool. Thanks, Nicole.
No problem.
I also put in the fish disease workshop, Western fish disease workshop that's happening in Tacoma in June.
Okay, cool. Thanks, Laura. All right. And
This
you
is
guys
a
always
reminder for everyone to remember to send these types of things towards Meg for the PNAMP newsletter. So just as she's gonna add the Patagus publication.
Yep, absolutely.
Good place
to share your information too, for sure.
Mm-hmm.
Yeah, we have definitely a larger scope with that newsletter. And so if you're not on there, let me know too. So if you guys are shy today, you can just send me an email and I can get everything out. And obviously the follow-up email, and then also in our newsletter if that's of interest to you, because it is definitely, it's a 3,000 plus group, so it goes everywhere. Alrighty, so we'll kind of move on since we're getting down to the wire. I'll just wrap up with a couple of reminders. After this meeting, like I mentioned like 15 times, I'll send out a follow-up email with links, the recording link, contact information for Mary and Zach, for me, for anybody that you want to talk to. If you'd like to be added to the distribution list, specifically the Fish Wiring Workgroup one, or if you have some interesting work you'd like to present, a presentation recommendation, just send me an email. We aim to be a resource for you guys, so just let us know if you have suggestions. And with that, we'll reconvene on October 16th, when we know everybody's field seasons are cranking up, so we kind of skip the summer quarter. To close out for today, I would love to give a big shout out to Zach and Mary for presenting their amazing work with Lamprey. I'd like to thank our core team, our task leads, and you guys, our work group. Enjoy the spring weather. It's beautiful in Portland today. And have a great day, everybody.
Thanks, Meg.
Awesome stuff. Thanks, everybody.
Thank you. We have snow.
What, Laura? No.
Just saying.
All right. And all my core team move over to the other meeting, and we'll do a quick debrief.
Thank you.